Yes, I must confess that I'm totally new to the EME field, but I'm learning an enormous amount about this absolutely fascinating disorder. But my task today, I was asked to introduce the Purinergic signaling field. Many of you won't have heard of it. This is basically that ATP is an extracellular signaling molecule. And there's a huge recent emphasis in this field in the pain field and in CNS inflammatory disorders. While there's currently no direct evidence that I could find for its involvement in ME, perhaps my talk might provoke some studies about this possibility, which may offer some novel therapeutic approaches to ME and CFS. What I'll do is start with some background information about purinergic signaling for those unfamiliar with this field, then talk briefly about purinergic signaling in the CNS and the autonomic nervous system, and finally talk about what is currently known about purinergic signaling in a variety of CNS disorders and in pain. So the story really started actually in the 60s when I was in Australia, when we discovered that there were nerves that were neither the classical adrenergic or cholinergic nerves, and then we looked into what the transmitter might be in these nerves, and it turned out that we had evidence that it was in fact ATP. I think there's one here. And this was the hypothesis that I came up with, and ATP is a purine nucleotide, so I invented the word purinergic for the signaling. So ATP stored in vesicles, released, acting on receptors, which we'll learn much more about in a minute, and it rapidly breaks down by ectoenzymes to adenosine, which is taken up by high-affinity uptake pump and reincorporated into ATP for further use. If the adenosine breaks down further to inosine hyperxanthine, they're usually inactive and they're whipped off in the circulation. I have to say that for the next 20 years, nobody much believed in it, and it was a pretty tough time. And it was kind of understandable because ATP is so well established at that time as an intracellular energy source. Why would it be such a ubiquitous molecule, an extracellular signaling molecule? And my wife wrote a poem, this was the beginning of it, a busy bee called ATP whose wings were full of energy, who worked and built inside a cell, emerged to see what he could tell to each and every other cell. The next conceptual step was to realize that ATP is in fact a co-transmitter in not only sympathetic nerves with noradrenaline, but as it turns out, this was not popular. I came from Australia to England, and it challenged Dale's principle, which was supposed to mean, was taken to mean one nerve only releases one transmitter. So this was the second unpopular idea that I had. But it turned out to be right, and it's now well established that ATP is a co-transmitter in all the nerves in the peripheral nervous system, parasympathetic, sensory motor, and so on, and also in the latest work together with GABA and glutamate and so on in the CNS. So it's a very important signaling molecule. And as we'll see in a minute, it's a very primitive, ancient signaling molecule. If you have a new signaling molecule like ATP, you have to find out what the receptors are for this. And the first step in that was in '78, when I realized that there were two different families for adenosine, which are called P1 receptors, and for ATP and ADP, which we call P2 receptors. Methylxanthines like caffeine and theophylline are very effective in blocking these receptors. They do nothing to the P2 receptors. The next step with receptors was in '85 when we realized on the basis of pharmacology that there were two subtypes of the P2X receptor, which we call P2X and P2y. And as we'll see in a minute, when cloning and characterization took place, this was confirmed and extended. For the P1 receptors, 4 subtypes were cloned and characterized in the early 90s, A1, A2A, A2B, and A3. And they're involved in a lot of different diseases, which I won't go into now. I should say that in the early 90s, when the cloning and characterization of these receptors took place, this was probably the turning point when people began to realize this was an important system. In '93, we characterized and cloned the first G-protein coupled receptors, P2Y1 and P2Y2, by Julius's lab in San Francisco. And then a year later, the ion channel, the first 2 ion channel receptors, P2X1 and 2, were cloned and characterized. To put it in broader perspective, nearly all the transmitters have both ion channel receptors and G-protein coupled receptors. For acetylcholine, of course, nicotinic, muscarinic, GABA A, GABA B, glutamate. And for ATP, then, we established P2X receptors as ion channel receptors, and 7 subtypes were cloned and characterized. And for the G-protein coupled receptors, P2Y, 8 have been established and characterized. It's interesting that for noradrenaline, there are no ionotropic receptors, and for glycine, there are no metabotropic receptors. I won't labor this, but these are the molecular structures of P1 and P2Y and P2X receptors. A lot is known about this, and people remove parts of it and find out what's involved. These are the subtypes of the P2X receptor, and it's interesting. The more I learn about this disease, the more I realize how many systems seem to be affected. And this turns out that the Purinergic signaling system is also involved in many. P2X1 receptors are very strongly represented in smooth muscle, bladder, gut, and incidentally, I've just heard that the sense organs are also affected in ME. There's a big involvement of P2X1 receptors in inner ear biology, and in the eye, and in the nasal epithelium, and in the tongue. P2X2, 4, and 6 are mostly involved in the central nervous system. P2X3, as we'll see in a minute, is fascinating because it's mostly on sensory neurons, particularly nociceptive pain neurons. And P2X7 is absolutely fascinating because it has an extra length of amino acids in the C-terminus, and that gives it another property. Not only does it open cation channels like all the others do, but it then opens huge pores which lead to apoptotic cell death. And this is very much involved in inflammation disorders of one kind and another, and also in cancer. We'll come back to that. P2y receptors, these are the subtypes, P2Y1, 12 and 13. It's mostly ADP sensitive. P2Y2, 4, and 6 are sensitive to pyrimidines. Again, you see they're widely distributed. I don't have time to go into that. In fact, although the early emphasis was on the nervous system and ATP as a transmitter, co-transmitter, we now know through immunostaining and RT-PCR for messenger RNA that nearly all cells in the body are absolutely loaded with the multiple receptors. In fact, it's a problem to work out how they interact with each other in their physiological roles. And immune cells are particularly heavily supplied by purino receptors, again, especially P2X7. which might be very important. I'll come back to this in ME. Special senses, fibroblasts, keratinocytes, and so on. I also should point out in the last couple of years there have been very interesting cloning of these ATP receptors in very primitive animals. In amoeba, in schistosoma, and even in green algae, the molecular structure of the receptors to ATP are almost identical to those in human beings, which means that this is a very primitive signaling system, perhaps the most primitive signaling system, and it explains why it's so widely spread throughout almost every system in the body. It's also, every time ATP is released, either from nerves or from other cells, it's rapidly broken down by ectoenzymes, and a great deal is now known about the various enzymes which break ATP down to ADP or to AMP, and then eventually to adenosine. The other interesting thing, people said, well, you've got all these receptors, where does the ATP come from? And initially, or for many years, people thought it was just from damaged or dying cells. It does come out from damaged and dying cells, but we now know that all these cell types, with the most gentle of mechanical distortion, release ATP without any damage to the cells at all. For instance, in the vascular system, changes in blood flow produces shear stress, which releases masses of ATP from endothelial cells, which then act on endothelial cells to produce nitric oxide and vasodilatation. Very important part of the vascular system. Another important conceptual step, again, was about 92, when there were papers published in Nature showing that Purinergic signaling was involved in synaptic transmission between neurons in ganglia and in the brain. And this is when the neuroscientists woke up to this system. The other important direction that the field has taken, we started mostly interested in short-term signaling in neurotransmission, neuromodulation, secretion, platelet aggregation. But later on, there's now a huge interest in long-term trophic control of proliferation, differentiation, and cell death in development, regeneration, neuroprotection, wound healing, and so on. We now turn to the brain, and in a developmental study, we showed that the first receptor to express early on in the fetus is P2X3, then P2X2 and 7, then 4 and 5, then 6 and 6, and only after birth does P2X1 start expressing. We now know that glial cells are very important, and all the glial cells have expressed lots of P2XY and P1 receptors, whether you're looking at astrocytes, microglia, or oligodendrocytes, or Muller cells in the eye, or Schwann cells in the periphery, or enteric glial cells. This is too complicated to go through, but there's a huge interest now in whole meetings on neuron-glial cell interactions in the brain, and ATP is very much involved in these because it's released from astrocytes together with glutamate, as well as a co-transmitter from nerves. There's a lot of growing interest in the involvement of purinergic signaling in normal behavior, in learning and memory, in sleep, especially adenosine receptors, but also ATP, in locomotion control, in feeding, and in mood and motivation. Anybody who's interested, I can give them more details about this. And then as for CNS disorders, all these have now, there's a lot of interest in purinergic signaling in trauma and stroke, ischemia, surgery. All the neurodegenerative diseases are being studied: migraine, neuropsychiatric disorders, mood, depression, anxiety, schizophrenia, epileptic seizures, and so on, which makes me think that it's worth exploring this system in ME. If it's involved in all these things, why would it not be also involved perhaps in ME? The first drug that came out of our work might be very relevant. It's called clopidogrel. It's a P2y antagonist for platelet aggregation, and it's used against stroke and thrombosis. And I heard yesterday that thrombosis is one of the features of ME, and so clopidogrel might be of some interest in relation to the treatment of that aspect of the disorder. Cancer. ATP is amazingly effective against cancer. We've looked at bladder cancer, prostate cancer, melanoma. Here's just one example. If you inject the prostatic tumor cell under the rat skin and then treat with ATP, it's extremely effective in reducing the size of the tumor. Of course, we've got in the brain neuroblastomas and gliomas, and these are also, there's a huge literature on purinergic signaling in reducing these. I came up with this idea in '96 in a paper to The Lancet about the involvement of ATP in pain. As I mentioned earlier, P2X3 receptors are strongly expressed on nociceptive nerve ending. So I suggested that for sympathetic pain, like causalgia or reflex sympathetic dystrophy, that it's not the noradrenaline that's coming out of sympathetic nerves, because noradrenaline antagonists have never been effective against sympathetic disorders, but guanethidine, which blocks the release of transmitters, stops the release of ATP, is often quite effective. So I think it's the ATP coming out of sympathetic nerves which is initiating the pain. For vascular pain, whether you're looking at migraine or ischemic muscle pain, which again is relevant to ME, I have this hypothesis. As I mentioned earlier, well, for instance, in migraine, a non-specific constrictor is not associated with pain, but that's always followed by reactive hyperemia, which is associated with pain. And with reactive hyperemia, ATP is pouring out of the endothelial cells. And my suggestion is that in the microvasculature, the ATP that's coming out of the endothelial cells can easily reach the nerves on the other side of a few muscle cells. And if these are sensory nerves, this would be a cause of pain. So we'll come back to the possibility that P2X3 antagonists might be effective against the muscle pain, which I gather is quite common in ME. And also, I had a hypothesis about cancer pain, which I won't go into now because of time. A very exciting and original finding from Japan about neuropathic pain came up that the microglia in the brain seem to be terribly important. They express in P2X4 and P2X7 receptors, and especially they're upgraded in neuropathic pain. This is a resting microglial cell, and these are the change in phenotype when it reaches damaged areas. It turns out that P2X7 and 4 antagonists really are very effective against neuropathic pain. The exact mechanism is not clearly known, but again, it emphasizes the importance of inflammation in purinergic signaling, particularly P2X7. Then there are papers now in Alzheimer's disease where again P2X7 but also P2y receptors are implicated. And this was a curious experiment we did years ago. We transplanted the nervous system of the gut into the brain. Why did we do this? This was in relation to Parkinson's disease, where dopamine is missing. And people that were using fetal dopamine, but there were huge immunological resistance problems and didn't work very well. But it turns out that the gut has got millions of dopamine neurons. And so we thought if we transplant our own bit of gut into the brain, we would avoid all those immunological problems. And we did this in an animal model, and they grow very well in there. But the surprising and interesting thing was that that bit of nervous system causes massive growth of central neurons, which notoriously don't grow very well. And the story there, to cut a long story short, is that it's the synergistic action of growth factors released from enteric glial cells together with ATP and nitric oxide which cause this massive growth. And we're looking at this whole thing in relation to spinal cord injury at the present time. But the idea that a growth factor works together with a small molecule like ATP is catching on, for instance, in stem cell biology now. This looks like a basis for how it works. There are a lot of ATP receptors on stem cells, and they work synergistically with growth factors to differentiate into different types of cells. Again, in Parkinson's disease, there's a lot of attention about the involvement and interaction of A2A adenosine receptors and dopamine D2 receptors. They probably form oligomers. Then in mood disorders, there are papers on mania and aggression, panic disorder, anxiety, depression, and schizophrenia. And loads of papers on epilepsy. And I found only one paper on ME, which wasn't awfully good, but it did show that that they claim that increases were seen in these CFS subjects with comorbid fibromyalgia syndrome, and were highly correlated with symptoms of physical fatigue, mental fatigue and pain. It's the beginning, I guess, and I hope there'll be more. Let me just finish by saying, well, what kinds of therapeutic approaches can you take with purinergic signaling? There are a lot of drug companies developing selective P2 receptor subtype agonists and antagonists. That's an obvious one. We're beginning to know what controls the expression of P2 receptors, and that might be an approach. Certainly looking for good inhibitors of extracellular ATP breakdown could be a way forward. And once we understand the ATP transport mechanisms, enhancers and inhibitors would be useful tools. And we must never forget about interactions with other systems. The latest thing, and these drugs are in clinical trials and are not exactly available, sadly, yet, but they will be soon, for both P2X1 and for P2X3 and 2,3, a heteromultimer, and for P2X7. The thing about these drugs, that's why they're in clinical trials, is that they're orally bioavailable and metabolically stable in vivo. A lot of the other antagonists and agonists are useful in vitro models, but they're no good in vivo. But this is the start, and it won't be long, I think, before these turn out to be useful and available. This is just pleases me. It shows the it shows the growth of this field. Just the papers on P2, not even adenosine receptors. You see, the first 20 years was pretty slow, but now it's exponential, and that's very exciting for me, I have to say. We've started a journal called Purinergic Signaling with a very international field, and it already has an impact factor well over 3 after 6 months. And this is my last slide. I am an old man, that's why there's so many names there. But first of all, great collaborators in Australia, and then when I moved to London in '75, lots more international people, many postdocs, great people I'm very fond of. I wish I could carry on for a lot longer. Thank you very much.